fisheries biologist at the Minnesota DNR, and I work with Corey Goldsworthy, who's the area supervisor for Superior, and I also want to acknowledge Derek Barr, who helped with a lot of the analysis. So I'm going to be talking about our efforts to sample spawning-sized Cisco using hydroacoustics. With hydroacoustics, just in general, there's 3 different components to the sampling that's all centered on the research objective and the species of interest. So these components include the survey design, when we go out and sample, how we space the transects, how much effort we put forth. We have another component, which is actually the physical data collection. So that includes things like how we calibrate the equipment and what sort of ping parameters we do, like ping rate. And then the third component is the data analysis component. And that includes, this is really related in particular to the species of interest. It includes things like setting up cell sizes and establishing thresholds and partitioning out targets. So in the Minnesota DNR, the original objective when we began hydroacoustic sampling in our agency was to estimate pelagic forage fish populations in the Minnesota waters of Lake Superior. And this was related to some of the goals with lake trout restoration, to look at the abundance of forage and to run some bioenergetics and look whether there was forage available for the recovering lake trout. So the pilot work with this project was completed in 2003 and 2004, and we basically gave all the hydroacoustic data to somebody externally to analyze during the years 2007 to 2012. So our objective here then was to estimate the biomass of all forage species in the Minnesota waters, and associated with that, a survey design was put in place, which is actually the survey design that we use through today. And we go out typically in August, and we have 3 different units. We have MN1 is the farthest south, and then there's MN2 unit and MN3. And the survey design basically has transects that are located both near shore, so in waters less than 80 meters deep, and then offshore in waters greater than 80 meters deep. And we have both near shore and offshore transects in each unit. And then associated with the offshore transects, we also have step trawl data off the blue heron. So we do step trawls from 70 meters depth deep up to 0 meters to get some fish data. So we had a survey design, and corresponding with the time that we began to use hydroacoustics in our agency, there was actually a group of people getting together that were writing a standard operating procedure for fisheries acoustics in the Great Lakes. And this Great Lakes SOP is really good for setting up the physical data collection, setting parameters for ping rates and pulse durations. Also, it's pretty clear on how you calibrate the equipment and how you analyze that data and then apply offsets. So the Great Lakes SOP, and I should mention too that the Great Lakes SOP is really pretty standard for all species, so it's not really species specific. So we have the second component here with the physical data collection that was put in place basically according to the Great Lakes SOP. And then the final component was the data analysis, which, as I mentioned before, we just handed off to somebody externally. But beginning around 2013, we began developing an inland hydroacoustic program. And so around that time, associated with sort of ramping up an inland hydroacoustic program and bringing on some people to analyze the data, we began to analyze the Lake Superior data internally as well. And associated with that, we sort of realized that this data set was being used in a new way. It was used primarily to look at spawning-sized Cisco and to use those estimates to set a commercial fishing quota. So revisiting this, then our objective had changed to really looking at just the spawning-sized Cisco and estimating biomass. So when we began to analyze the data internally, we realized, well, our objective has changed. It's important to revisit, first of all, how the data is being analyzed, and secondly, whether the survey design is appropriate for sampling spawning-sized Cisco. So we had a large time series of data, and the first thing we wanted to do is look at that and look at the data analysis and say, are we analyzing this data in the best way that we can for spawning-sized Cisco? So associated with that, we developed a standard operating procedure to standardize the data just for those larger Cisco. And we went through a series of steps to develop this SOP. We looked at setting a Cisco threshold, how to deal with scatter from large targets that were unlikely to be Cisco, how to convert the hydroacoustic density estimates to biomass estimates, how to model and extrapolate that up because we needed a population estimate for all the Minnesota waters in order to set a quota, and then how to archive and manage the data. So I'm just going to step through each of these really quickly and briefly and show the process by which we kind of made these decisions and moved ahead with the SOP. So the first thing we needed to do is to determine a threshold, and we wanted to do that for spawning size Cisco. And what we see with the commercial take is that typically fish are caught between 300 and 500 millimeters in length. When we looked at the trawl data corresponding with when we did our hydroacoustic surveys, we saw that there was a really nice separation between Cisco and all other deep water Coregonid species such as bloater and kai, and also with rainbow smelt. So we felt that we could use the hydroacoustic data, we could use the in situ data, and try and find this break point for Cisco. Instead of typically in hydroacoustics, single target data is used. We actually use fish tracks, and we did that partly to eliminate a bias. Typically, due to the way the beam, the physical characteristics of the beam, near the surface, the sampling area, the sampling volume is much smaller, and it gets wider as you go deeper. So targets that are deeper tend to be the beam longer and return more single targets. So in this case, we had 11 single targets, but fish tracks really identified 4 different fish. So it removed the bias of these deep fish that were in the beam longer and returned more single targets. And then we used a cluster analysis, and we found a breakpoint in the data that was around negative 35.2 decibels. And that corresponds pretty well to a negative 35.6 threshold that Yule et al. Identified in his 2006 paper when he was looking at spawning size Cisco. And using sort of a standard equation, Rudstom's 1987 equation, negative 35.2 decibels is about 290 millimeters in length. So that works out really well. That corresponds pretty well to that 300 millimeter minimum that we saw from the commercial data. The next step we did is we wanted to remove some of the variability and bias from large scatterers that were unlikely to be Cisco. So we removed backscatter from targets that were greater than negative 30 decibels. This works out to be about 500 millimeters in length, so sort of that upper side of the fish that are harvested by our commercial fishers. So we did that by physically removing fish tracks that were large, and also looking at the density or the biomass data and looking for outliers. And if we found an outlier, we go back to the data, we look at it and be like, okay, well, this looks like it's coming overwhelmingly from one fish. It's probably something other than a Cisco. So that removed some of the variability in the data as well. To convert density to biomass, we basically took the average target strength and converted that to length using Rudstom's equation, and then from length to weight using an equation published by Yule in 2013. This graph, I don't know if I can use my pointer, but this graph over here, the white dots are the Minnesota DNR trawl data from 2012 to 2013. You can see that the relationship fits our data really well. The only reason we used Yule's relationship rather than developing our own is because we didn't have a lot of fish greater than 400 millimeters in our trawl data. So the next step then was we wanted to model the data and then extrapolate it to come up with a population estimate. The way we did that is we plotted out the biomass by bottom depth, because sometimes particularly near shore, there's a relationship where the deeper the transect, the more biomass of fish we saw. And then we fit a relationship to that. And then we incorporated autocorrelation between neighboring cells using an autoregressive moving average statistical process. So we used the model then to extrapolate out an average biomass to the volume water at that depth, at that location. We got an average biomass, and then we used that model that incorporated the autocorrelation to bootstrap confidence intervals. And then lastly, we wanted a way that we could organize the data so it was transparent, so multiple users could look at it, we could see the decisions that were being made. So we developed a data management system. We had templates for the analysis files, we archived the data so it was accessible by multiple users. We developed a database with links to the analysis files and also that documented all the steps, so that was a very transparent process. And then we analyzed the data in R with output files that could be easily re-imported if we wanted to go back and reanalyze the data. So I think an important part of developing SOP is to see whether it works. And we had 2 different independent users analyze the entire year of data and found that using the SOP that the differences between users was pretty minimal, so it was less than 5%. And we felt that was pretty good. And doing this exercise also helped us see where there were potential differences and where there might be a potential for more standardization. So at this point then, we have our new objective, which was to estimate biomass of spawning size Cisco in the Minnesota waters, data collection, which was previously kind of set by the Great Lakes SOP. We felt that we'd taken some good steps to standardize the data analysis for spawning size Cisco, and that allows us to kind of reevaluate or revisit this idea of survey design. So since we have the data analysis, we can go back through the whole time series of data we had, we can look at the trends, and we can look at that in comparison to some of the other data we have as well. So we have a hydroacoustic time series of data that's been collected since 2007, and we actually applied our standard operating procedure methods, but we tweaked it a little bit just to kind of see how robust were our estimates and how much does changing things slightly have an effect with overall estimates. So the 3 methods we used is we applied a high threshold for Cisco, and I mentioned before that we applied a negative 35.2 decibel threshold, but the Great Lakes SOP recommends backstepping that 6 decibels when you're analyzing the total of the backscatter data. So that's why it's negative 41.2 here instead of negative 35.2. But we did down to 75 meters depth. And the reason we went to 75 meters is because we had step trawl data from 70 meters up to 0, so we don't really know the species that are below that depth, so we just excluded it. The second analysis method was again to use that higher threshold for Cisco, but only to go down to 35 meters depth. The reason we did that is when we plotted out the fish tracks, we saw, again, here's our negative 35 threshold for spawning size Cisco. It looked at, like, beyond 35 meters deep that there was this concentration of fish that were smaller than Cisco. What this says to me is that there's a potential change in species composition as you move deeper into the water column. And potentially when you backstep the threshold for analyzing or integrating the backscatter data, the SV data, we had some concerns that maybe we were starting to integrate a bunch of backscatter from some of these fish that were unlikely to be Cisco. So we thought, well, we could just cut off everything below 35 meters, and this would represent sort of a minimum estimate for spawning size Cisco, assuming that they're not found a graded one that's below 35 meters. And then lastly, we applied a very low threshold, so a negative 66 decibel threshold, which incorporates the backscatter from all species. And we went down to 75 meters, but we partitioned the targets out by depth. So again, here's our cutoff for spawning size Cisco, and we looked at 5 meter depth increments and the portion, or we basically set up a ratio, how many spawning size Cisco in each of those depth intervals versus all other fish. And this method is very similar to what Daniel does when he analyzes Lake Superior data. So the time series, as I mentioned previously to 2013, we sent out the data to be analyzed externally. And so these green bars represent the data that's pre-SOP that was analyzed externally. And then we have our standard operating procedure methods. And again, we have those 3 methods. So the high threshold down to 75 meters, the high threshold down to 35 meters. So this represents, on average, about 10% less biomass than analyzing it all the way down to 75 meters. Again, sort of a minimum. And then that low threshold down to 70 meters, but partitioning out the targets. So in general, we see good correspondence between the 3 SOP methods. You can see that the pre-SOP is a little bit of an outlier. I should also mention that in 2014, we collected 2 data points. So we went out and did a summer survey that had been done previously. And then we also went out in October, collected some data, and in general, the October estimates were a little bit higher. So what does this mean in terms of setting a commercial quota? Well, Minnesota sets its quota by taking 10% of the lower 95% confidence interval for each year, and then taking an average of that from the previous 3 years. So this is what the quotas would look like from 2010 through 2014. Again, you can see this pre-SOP method is a little bit of an outlier, but even in terms of there's pretty good agreement between the 3 SOP methods, but even at that, the difference between sort of the high estimate and the low estimate ranges from about 10 to 20% each year, which isn't insignificant when you're going to a commercial fishing group and you're talking about changing, potentially you could harvest 20% more fish. So we can look at this time series in relation to some additional information we have. Mark just mentioned that they do a year class index with their trawl data. So that's the green bars here. And the Minnesota DNR in 2013 looked at the fish that were harvested in the spring and the fall fishery and aged those fish, and in general corresponded really well to where they saw the peaks with year class strength index from the USGS. And it takes about 4 years for a fish to recruit to spawning size Cisco. So again, here's our time series with just the 3 SOP methods. And so we saw in probably the past 15 years the largest blip in year class strength in 2003, which we'd expect to be fully recruited to spawning size by about 2007. Again, we saw a little tiny blip in 2005. We'd expect those fish to be fully recruited by about 2009. And then in 2009, we had another little blip, and we'd expect those fish to be recruited by 2013. There's a trend here of sort of decreasing abundance and then increasing abundance, with more recently kind of a peak around that 2013 year. Again, I put the 2014 data on there. Those estimates seem to be a little bit higher in the fall. In general, I think we would expect maybe this makes some assumptions about that the number of fish recruited into the fishery would be consistent with the year class strength. But if that were to be true, I think we'd expect to see the highest abundance in 2007, and we didn't necessarily really see that. We can also look at the hydroacoustic trends compared with our catch data. So again, we have this decreasing trend and sort of an increasing trend. We can average out the 3 SOP methods to get sort of an average acoustic trend, and we can compare that with the catch data. So this is commercial catch data from November. So this is the roe fishery from all the 3 Minnesota units. And we can also look at the summer commercial catch as well. But the hydroacoustic trends don't seem to match very well with what we see in terms of commercial catch. This is potentially, could be a spatial and a temporal mismatch. I think it's important to recognize that the commercial catch is more of an integrated sample over time, whereas the hydroacoustic sampling is really a snapshot. But another issue is potentially this idea of extrapolation. So when we go out and sample with hydroacoustics, we have density estimates from certain locations, but we need to extrapolate to areas that we haven't sampled in order to come up with an overall population. And we do this volumetrically, and in the Minnesota waters, MN2 offshore and MN3 offshore comprise the majority of the volume of water. And so they overwhelmingly contribute to the Cisco spawning estimates. So if we want to just back up for a minute and say, okay, we don't want to extrapolate, we just want to look at the density trends and try and match those up to catch data by location to try and potentially remove this effect of extrapolation and to make a better spatial match. We can try and do that as well. So we have here MN1 near shore data, the density estimates and number per cubic meter, the 3 SOP methods, which on average create a trend that looks something like this, compared to the Minnesota November catch data, and then the summer catch data. The summer catch data has been pretty consistent over time. You can see there's been a pretty big drop off with the roe catch. But neither of those correspond very well with the hydroacoustic trends we're seeing. And again, I want to emphasize here that hydroacoustics represents a snapshot, and in 2012, we had a very high abundance of large targets near shore. And we saw this also in MN2 near shore, which is an independent sample compared to MN1 near shore. And that's just the effect of going out and taking, sort of the snapshot approach. All other years, we saw the majority of the large targets were offshore, so it might just have been sort of an aberration year. We can also look at the offshore data, maybe make some assumptions that near shore the data is more variable, offshore maybe provide a better picture. Again, we can kind of look at the acoustic trends and compare that to our catch data. But in general, we don't see really good correlation. And we looked at the other units as well, MN2 and MN3. We also didn't see really strong correlation between hydroacoustic densities and catch data. So another important consideration is that there's potential for a lot of variability when we do this. We go out, we take one transect offshore, and we assume that's representative, and we hope that there's not a lot of variability. But we have a small data set to look at it. In 4 years, we actually did 2 transects, so we did one off the Blue Heron, and then we did a separate one with the DNR vessel. It wasn't the exact same location, but it covered the range of depths that we sampled with the Blue Heron. And in those years, we didn't see exactly what we hoped we would see. In 2012 and 2013, there was good agreement between the density estimates for MN3 offshore with the 2 boats. In 2009 and 2011, they were significantly different. So this says to me that we don't have a very good handle at this point on how much variability is associated with some of these transects. I should mention that these were averaged in order to come up with the overall population estimate. So to come back to this idea and revisit our idea of survey design, I think there's a lot of important considerations with hydroacoustics when we're setting up sampling design. And I think certainly timing is one of them, and I think this is a challenge for some of our agencies because there is both a flush fishery and there is a roe fishery, and potentially assessing the flush fishery and the roe fishery might mean sampling at different times. There's also issues associated with sampling effort. Certainly we're trying to, Lake Superior is a huge lake, and it becomes difficult. You have to weigh how much effort you want to put out versus cost, and try and develop a strategic way to locate the transects, to space them, and basically come up with an effective sampling plan. There's also some issues, and Mark talked about this a little bit, but currently with our current methods, we don't sample the near surface area very well. Given the physical properties of the beam is much smaller volume of water at the surface, so we just don't sample fish very effectively. Additionally, there's potential for boat avoidance issues, and there's also near field associated with the transducer, so there's a range beyond the transducer where we can't sample fish very effectively. So in general, we don't get any targets or very many targets less than 7 meters deep. This is where our commercial fishers target Cisco. So we're potentially not sampling the part of the water column where they're targeting the fish. And I think that's an important consideration issue to look at moving forward. And then again, this idea that the community composition could potentially change by depth. If we understood that better, we could potentially analyze the data more effectively. And finally, this idea of extrapolating to unsampled areas. So when quotas are set, we're trying to get a population estimate across all the waters in Minnesota, but that means that we have to extrapolate to areas that we sample to areas that we don't. And to do that very well, we need to have a better handle on variability. And I think this is also an important consideration for potentially if you were to move more towards like a November stock assessment where you go out when they're spawning in November. We know that that's the point at which Cisco are most highly concentrated as well, and they're very patchy. And I think it becomes very difficult to know how to effectively extrapolate when you're sampling a really highly concentrated patchy population. So to finish, I just want to emphasize that hydroacoustics is a sampling tool, and like all tools, there are both advantages and limitations. And I think moving ahead, that understanding the limitations of hydroacoustics is important for evaluating its effectiveness in terms of assessing populations. Unfortunately, it's expensive and time-consuming to evaluate survey design, but I think if there's multiple agencies that are looking at using hydroacoustics to assess their Cisco stocks, a coordinated effort could potentially provide answers much more efficiently. And then finally, I think it's important to emphasize that hydroacoustics is just one line of evidence for evaluating the status of the fishery. We're fortunate that we have some additional data, and I think combining abundance estimates from hydroacoustics and potentially even from the USGS trawls, along with commercial catch data and biological data like year class strength could potentially be a more robust method for setting quotas in the future. So we do have kind of a cookbook SOP that we're developing, and if other agencies are interested in taking a look at how we do it, feel free to contact me and I can get you that information. So I'd be happy to take any questions.